Hello everybody, my name is Michael Byun, a fourth-year food and human nutritional sciences student at the University of Manitoba. My project this past summer was about ginsenoside treatment of endothelial cells and whether it can mimic the vasodilatory response induced by G protein-coupled estrogen receptor agonist G1. My project was centered around atherosclerosis, a progressive cardiovascular disorder that leads to severe outcomes such as heart attack and stroke. This condition is associated with arterial stiffness and endothelial cell dysfunction, both of which are due to alterations in the endothelial cells that line the inner surface of blood vessels. From Figure 1, you can see that production of the potent vasodilator nitric oxide is upregulated by the signaling initiated by the G protein-coupled estrogen receptor. My project ultimately had two objectives. One was to conduct a literature review of the work surrounding GPR's role in early atherogenesis, outlined in Figure 1. And the second was to study and compare the vasodilatory ability of ginseng-derived ginsenoside, or RB1, to mimic the effects of GPR agonist G1. First, we wanted to establish that GPR is expressed in the EAHY926 cell line. Figure 2 highlights the use of Western blotting of confluent cell samples. This method uses antibodies to measure the amount of specific protein, in this case GPR, in cells under experimental conditions. Notably, the expected band of 55 kilodaltons documented in the literature was not observed in Figure 2a. Instead, 3 sets of bands of higher molecular weight were detected. Studies have previously shown that GPR has many glycosylation sites, which may explain a higher molecular weight, though ultimately further examination will be needed to be conducted. Next, we wanted to identify the concentrations of our compounds G1, G36, and RB1 that do not display toxicity in our cells. After reaching confluency, the cells were treated with the compounds as well as a vehicle control at the concentrations indicated, and images were taken with a EVOS cell imager. As shown in Figure 3, there were no observable signs of toxicity, such as abnormal cell morphology or free cells, for any of the treatments. This confirms the concentrations of our compounds that were selected are suitable for use in future experiments. From these results, future work would need to confirm the presence of GPR in our cell line. Next would be a time course experiment to study a temporal expression of vasodilatory related proteins, for example, PENOS, which produces nitric oxide, and vasoconstriction related proteins such as the angiotensin II receptor type 1. In conclusion, my project has developed a strong foundation for moving forward. The literature review highlights key regulatory mechanisms in which need further exploration, while my toxicity experiments make future studies feasible. The significance of these results, however, ultimately pertain to the promotion of further research done on natural relaxants to treat cardiovascular diseases such as atherosclerosis. Thank you.